Before we move on to our next topic, I just want to circle around and talk about our goals for this chapter. First of all, we want to talk about how lipids are assembled, and that means taking various components and putting them together with different types of linkages. And I'd note that our two most important functions for lipids are fuel and forming membranes. Now, let's remember that we moved on and talked about fatty acids and triacylglycerols as the fuel components. And where we'd like to go next is to talk about glycerophospholipids. And it's worth noting that those are membrane components. And I'd note that glycerophospholipids are part of the polar lipid category. That means they're amphipathic. And that's going to be a very important thing. When we talk about membranes, we want to think about amphipathicity. So let's go and review how we are putting components together. For fuel molecules, we said we took a glycerol backbone and 3 fatty acid groups, which were then esterified or linked by esters to that glycerol backbone. And this cartoon was a beautiful way to think about that because it showed the backbone and the 3 fatty acyl groups now connected by fragile ester linkages there. That cartoon is a great place to start for our next chapter. Let's go on to this page. Make a note that the title for this page is Glycerophospholipids. And let's make a note that glycerophospholipids are membrane function. Now, if we start again with the cartoon, what we can see is we have something very similar. We have a backbone, in fact that's a glycerol, and we have 2 fatty acids that are attached by ester linkages in the same way that we started with triacylglycerols. So here's how it works. We're going to take a glycerol backbone, which we're showing over here without the H's for the alcohol, and we're going to make a diacylglycerol instead of a triacylglycerol. Doesn't matter how many carbons we put on to these fatty acid chains, or these fatty acyl chains I should call them. We'll just put that like that. And what we've done, again, is created a diacylglycerol. Now, instead of having the 3rd alcohol of the glycerol become esterified to a 3rd fatty acid, we're going to attach something that's very polar to it. In fact, we're going to flip that alcohol to the left and we're going to connect it to a phosphate. So careful with the phosphate. It's tricky to draw for students who are not used to it. Number one, the phosphorus has 5 bonded pairs of electrons around it. It doesn't obey the octet rule. Second of all, it has 2 negative charges, although sometimes we might leave a proton on there and leave it as 1 negative charge. I usually draw it as 2 negative charges if it's just a phosphate added on there. Now, let's take a look at this and make sure you understand that this is a very amphipathic molecule. We learned about amphipathicity in our first review chapter, and the whole idea was that you had a polar head, and the more polar, the better. This has 1 or 2 negative charges. A full charge is always going to be very polar. And then we have 2 nonpolar tails, and these are very nonpolar. And we've got a classic amphipathic molecule here. This is called phosphatidic acid. Sometimes we refer to it as phosphatidate because it is a negatively charged conjugate base. But phosphatidic acid is a membrane lipid. In fact, it's one of the most popular ones. We sometimes abbreviate it as PA for phosphatidic acid. So the important thing is that we've created 2 esters. See that? CO, C double bond O. There's another one. And that 3rd one is not connected by a normal ester to something else. Instead, it is connected by a phosphoester. Look at that. C-O-P double bond O. Instead of C-O-C double bond O, it's C-O-P double bond O. And that is so important that we are going to write that down because it's going to be used every chapter from here to the final exam. That linkage is a phosphoester. This linkage is a regular ester, as is this one up here. So what it is an alcohol and a phosphoric acid. Take away a water, and you end up with a phosphoester. And we can see that with the cartoon out here. Nice description showing the glycerol backbone, two fatty acids that are esterified, and then we have a phosphoester out here to make our glycerophospholipid. However, notice there's something slightly different out here. See that? That means this could be an O minus, like it is with phosphatidic acid that we just drew, or we could have something else attached there. In fact, let's go on and look at the 4 other glycerophospholipids that I'd like you to be aware of. If we attach a choline group, let's make a note of that. If we attach a choline to that group, to that phosphate, we end up with a phosphatidylcholine. If we add an inositol group, we end up with phosphatidylinositol. If we add an ethanolamine, we end up with phosphatidylethanolamin. And lastly, well, let's give you a little test. What is this thing right here? This head group, what is it? Just remember that what we've done is we've taken an alcohol and buried it in this phosphate. And what that means is this is actually an alcohol if we were to liberate it. Pause it for a moment, see if you can recognize what that structure is. Okay, I hope you had a chance to look at it. And what it is a serine. And what you have here is the alpha carbon, carboxylic acid, NH3 over here for the amine. Here's the side chain, beta carbon, then an alcohol buried in this group over here. So this would give us, as you might expect, phosphatidylserine. Now, a couple more things before we leave this. I first want to say again something extremely important. If we have a CO, P double bond O, as we do, coming off that glycerol carbon there, that's a phosphoester. But notice we also have a COP double bond O coming from that direction. So that's another phosphoester. And this is a very important new linkage that we're going to talk about. If you have 2 phosphoesters that are converging on one phosphate, we have a special name for that. It's called a phosphodi for 2 ester. So it's not a phosphoester anymore, it's 2 phosphoesters buried into the same phosphate, and we're going to see that in this chapter, but we're also going to see it in the next chapter. So lastly, let's just make a note that, as we said for phosphatidic acid, which was PA, we sometimes call this PC, PI, PE, and PS. So we need to understand the components and the linkages. Let's finish it off this way. Glycerol backbone, fatty acid, both with esters, standard carbon esters, attached to the glycerol backbone. A phosphate attached by a phosphoester, and a head group such as serine or ethanolamine or the others attached by another phosphoester, and the 2 phosphoesters together make a phosphodiester. A bit complex, but now you know how to make the components of lipid membranes, and that's really important for biology and moving forward.